last several years, we've seen some strange genetically modified organisms. Pigs that glow in the dark, chickens that grow without feathers, and even goats that produce spider silk proteins. But what really gets people riled up are genetically modified foods. The prospect of eating somebody's science project turns the stomach of those who aspire to eat so-called whole or natural foods. But what exactly does genetically modified mean anyways? And how are those foods created? To answer these questions, we need to go back in time about 10,000 years. That's when people first started domesticating animals and cultivating plants for food. By picking organisms with desirable traits and mating them together over and over again, people were able to mold species to meet their dietary needs. The classic example is corn, which started out as a tropical grass called teosinte, with small, nearly inedible kernels. Over thousands of years, ancient farmers in Mexico transformed the grass into the delicious, starchy treat we like to eat today. We now know those ancient farmers were manipulating the DNA of the teosinte plant. As few as 5 changes to its genome, the long strands of DNA that encode for proteins, create the dramatic differences we see between teosinte and corn. Scientists call this process artificial selection, and it's made all kinds of unappetizing plants edible, from wheat to rice to almonds and bananas. What's different about modern genetically modified foods is the speed and precision with which we can make changes. Now, instead of selectively breeding for traits, we can go directly to the DNA responsible, snip it out, and transplant it into newly developing plants and animals. If these snippets come from the same species, scientists call the new organism cisgenic. Cis means the same. But if the DNA comes from a completely different plant or animal, scientists call the new organism transgenic. Trans means to cross. Those fluorescent pigs, they glow green because they contain a gene from a luminescent jellyfish. And in the last 20 years, we've begun to see the food equivalent. Corn that makes bacterial toxins poisonous to pests, tomatoes that make the antifreeze proteins from fish, and even cows that produce human milk. So you may be wondering if this kind of overt manipulation is safe. Well, all of the GM foods currently on the market have been thoroughly tested, but the verdict's still out on some of the more exotic transgenic pairings. Personally, I'm waiting for a tomato that I can cut in the dark. For Scientific American's Instant Egghead, I'm Eric Olson.
Wow, grade 12. Good morning. And wasn't that an interesting story? Sometimes I think, is it really true? But it is true. And that story of the Millie that came from a quite normal veldgrass, I think this is extraordinary. And this is just telling me how much power God invested in the human being. We are so great. But what can we do with that? So, we can also alter the DNA of plants and animals. And then we say it is GM organisms. GM means genetically modified. An organism that has a change, like the long-life tomato, is a GM organism. Now, and a GMO is that genetically modified organism. That's what it means. When we look at plants, they want to manipulate the genes of a plant to make it more pest resistant, especially viruses, so that it is not easy for the virus to attack the cells, and also insect resistant, in such a way that the insects are not interested in eating the plant, use the plant as a space to lay their eggs in. You know, the mealie farmers, they really have troubles with an insect that is laying its eggs inside the cob. And then it's actually using that inside, respirating in there. It's becoming a very acidic smelling inside, and they cannot use that cob then, and there's no mealies to sell or to give to their animals. So pest and insect resistant. That is how they try, and they are succeeding in plants. They also want to change the crops into more drought resistant, so that it can wait a bit for the rain and still stay alive. Pest resistant, drought resistant, insect resistant. Also, the crops resistant to herbicides. Herbicides are weed killers. Why? The thing is, when you plant your crop, you put in a lot of fertilizer and water, and the environment is perfect. Now the weeds, those little plants, the veld plants, they can also grow and use all this fertilizer and water. If you spray it then with a poison, you will also kill your crops. You remember that specific weed killer that only kills the broadleaf, the dicotyledon plants? That was where they use auxin as a herbicide. You remember that? That was part of our growth stimulant chapter. But here, they want to change the crop so that the crop is not affected by the herbicide. Interesting. Then, by manipulating the genes, it can improve the quality of the plant. Maybe more juicier, sweeter, bigger. That can help. There we see the mealie, some crop that is growing there. And then, of course, bananas, all the fruits. A very interesting case that happened in the East, where rice is their staple food, is that they change the normal rice, which is very high in vitamin B. And vitamin B is a very important vitamin, especially for the nervous system, but they also need some other vitamins. Yes. So I want you to guess which vitamin has this golden, and if I say orange color, what will you say? Yes, vitamin A. Vitamin A, carrots, beta carotene, is the substance that now can be produced inside the rice. They call this now a golden rice, and it is actually now enhanced with also vitamin A. And if we think, not again, take from wherever you want the gene, in this case a bacteria, include it in the cell of the plant you want to increase. Then, with culturing on agar, make it vegetative or asexual reproduction, increase this little cells until this little cells can form roots, and there I have a plant with the new gene. Allow this plant to breed and make lots of little plants like that. Question: this plant, if it is breeded, will it be sexually or asexually breeded? Asexually. Why? Because it's a fast way, and it ensures that all the plants has this new gene and this new way of expressing its genes. Does it make sense? I really hope so. Create recombinant bacteria with the desired gene. Allow the bacteria to infect the plant cells. Desired gene is inserted into the plant chromosome. Make sure that the plant is cultivated. We call it micropropagation. Also back in a previous chapter, micropropagation means asexual reproduction. Take a few cells, put it on a cultivating medium like agar, wait till there's enough cells, it forms roots and plant. Allow this plant to grow and become strong. Now, the potential problems that arises from GMOs and that worries people are, if you are belonging to a cultural group, a religion, or you are a vegetarian, and you are not allowed or do not allow yourself to eat genes from a specific animal or any animal, then you will not know or you will not eat some vegetables anymore, because transgenic, where the gene of an animal is put into a plant in order to get a specific feature. Long life tomatoes came from a fish. Yes, they walked down the beach and saw dead fish. Tomorrow, same dead fish, but no rotting takes place. This is a plan. Let's get that gene that keeps this fish from rotting. Rotting? Being broken down by bacteria. And they use that for long life tomatoes. Please don't think that long life tomatoes and long life milk is the same thing. No, completely different. Okay, let's continue. So the first problem is you don't know which organism's gene was planted into the plant that you want to eat. Second thing, transfer genes can cause allergies. Is there enough research? And that's, I think, another point. Is there enough research to see what is the effect of genetically modified food? They are very much worried that they will make super weeds, because now you make a plant and this plant is resistant to a weed. But what if there is a crossbreeding between your weed and this plant, and now we have super weeds that take over the harvest, and unfortunately lots of money goes into making new poison. Now, because weeds, just as mosquitoes and other insects, can become resistant. And that is our point number 5. Resistance can change. And now you will have to come with a whole new plant if this weed that was that was killed by the herbicide now can grow through that herbicide. And it takes a lot of money. New pathogens can arise that we have no vaccines or toxins for. Antibiotic resistance, the disease now can spread, and not only antibiotic resistance, but resistance to this newly formed herbicides. Perhaps if we think the dicotyledons that can grow through the auxins and become stronger now. And then it can be used in biological warfare. biological warfare when the super pathogens for a biology student come and conquer human cells, specific animal cells. And there's lots of stories of a conspiracy with this corona. I do not want to go in this, but we actually experience now what can happen to the whole world if there is a virus or any other type of pathogen that we do not have an answer or a weapon for. And then, as I have mentioned, it is costly. It is costly first to do the genetic modification, because you have to do a lot of research. It is not just tarago, and there I have the piggy that shine like fluorescent, or whatever I'm trying to accomplish. It is not like that. It is many hours of trial and error. Then also to test, is it safe? Is it good for the environment? If we look at superweeds, that will not be good for the environment, because some plants will take over the good plants that is food for human, but also plants that are food for other organisms in the food webs. Now, and that costs a lot of money, so we have to take care before we just consider this as the all answer. So what do you think about eating genetically modified food? I would love to hear your opinion on this. Very nice cartoon when we have definitely two scientists and they have two breakthroughs. Oh, they are so excited. The one is imitation eggs made of soybeans. Bravo. But the other one is imitation soybeans made of eggs. Okay, why not just rather eat eggs and soybeans? But that is perhaps how some people feel about that. Right. Now the human genome. The genome of the human means when we can have a chart or a table with every single existing gene that exists in the human race. That is the human genome. Every type of hair color, eye color, or the mutations that happened, every single one. And do yourself a favor and just type in your Google search and say eye colors, and you will see how many are there. But that is the Human Genome Project. They want to map the base sequence to know which nitrogenous base sequences is responsible for each gene. Now, they want to identify all the genes of the human. They want to know the nucleotide pairs for all the genes. And it is interesting that there are sometimes where more than one pair have the same outcome. Okay? Gene expression. What is the protein produced by a specific gene code? And then the role of each gene. That is the Human Genome Project. And here is just a very simplified diagram to show us how they started to see what is happening and what is each little part on the chromosomes telling us. So why do we want to do that? We want to do that to understand genetic diseases. You remember those X-linked diseases, the mutations like hemophilia that is not an X-linked, but it make a lot of people suffer, genetic diseases, to study them, to prevent them from recurring, and also how can it be treated and to improve treatments. A very interesting one that I came across was cystic fibrosis. You remember that cystic fibrosis is where the mucus are very sticky. What happens with the sticky mucus is that the chlorine ions inside a cell becomes too much, and that cell starts to change the way it's behaving. And the sticky mucus prevent the absorption, the diffusion of oxygen, really difficult to breathe. It also causes the pancreas ducts to become clogged and blocked. So, the treatment uses a type of virus called a lentivirus to deliver a healthy copy of the gene, the CFTR gene. So it is a recessive gene that causes the sticky mucus. Now, a virus will go into a cell. The virus change the DNA of a cell to make more viri. You remember that? Okay, so viruses, and now they take that. It causes the cystic fibrosis. When it carries a mutation, the gene therapy will be given by inhalation to better target the cells. And this was in 2017. I have read about this. I wonder what happened. Is it really working? But this is how they try to help a genetic disease. This part is not part of your syllabus, but it is so interesting. With gel electrophoresis, it is the same as chromatography, where they can identify DNA. They have it's looking like a car battery, but it has cells full of gel. They inject the DNA into the gel, and then it forms bands. You do not have to know exactly how it happens, but this is the gel make the fragments separate from one another in each tube where it is injected to. And this can help us to show a specific DNA structure. So they take a photograph of that and they keep it, like your fingerprints is telling you who you are. But when you are baking a lot or cutting yourself a lot, now you can damage your fingerprints and later there's no fingerprints left. But this cannot change. So there they have the bands labeling or showing the DNA. And then they already use it for crime, searching for a criminal. This is the DNA that they got on the crime scene. Now they had some suspects, and who do you think is the guilty one? We can look at the DNA and see that these are similar to the DNA found on the crime scene. Interesting. Now, and this is also a very interesting job or career that you can think of, where you are looking into criminology, but you use your biological knowledge. You can go and help to take off all the little maggots from a corpse, and then you can date how long is this person dead. You know, all this CSI programs? Yes, that they need a biologist for. Or you can become a genetic engineer where you study the DNA and you help with altering people's lives, making better food. I hope that you enjoyed this very interesting piece of work. Please complete your worksheet just to see, do you really understand this new chapter? And remember, I also added the answers to the previous worksheet. Please mark your work, do your corrections. I think this is so interesting that if you have studied this once and go through this with a lot of concentration, you will remember this, grade 12s. You are so sharp. So please do that. Do not think, I will do it later. Do it now.
Namibia is known for its beautiful wide open landscapes. However, these deserted spaces also pose numerous educational challenges in our country's rural areas. Because of this, many learners are denied access to proper teaching and learning. At Eduvision, we believe that education is the starting point to a journey of endless possibilities. It is Eduvision's goal to prepare and guide learners in order to make the modern world they are living in accessible to all.
Accommodation is the ability of the eye to focus on distant and near objects.
And I bring it in contact with the electroscope. What you must keep in mind is positive charge can't move. It is the negative charge that moves.
The first one, EF, is then going to be 0 minus negative 2 over 4 minus negative 4. Look at my signs. If you go wrong there, if you don't substitute in brackets, you're definitely going to get wrong answers.
One day we went to I think it's the opening of Oshakati Stadium. Then I was a lead singer there. Then this DJ Remind came to me and was like, "Hi, don't you want to be a solo artist?" I said, "What is solo?"
Ladies and gentlemen, my name is DJ Siambra Mazgrum Kwampiri Perez Kabuka Pleasure Return from the Mighty region.
Happy Friday, what's up? Easy Friday, happy Friday.
Abdallah is joining us in the hot seat. She said it right. She said it right. She said it right. Welcome to the tribe.
Thank you so much.
Hi to Holland, Namibia. Welcome to the tribe on One Africa TV. My name is Shay the Goddess, and I keep you company every Friday along with some of Namibia's biggest artists right here on this platform.
Share with us on #LearnOn1. Tell us what you're learning and where you're learning it from. Send us your name and town to the One Africa TV's WhatsApp on 081 200 6659 or send an SMS to 555. One Africa TV. It just gets better.
Our 31st new chapter, and that brings us one chapter closer to completing our course of this very interesting subject that is all about life. Genetic engineering. What is the function of an engineer? An engineer is someone that can make plans. An engineer takes new stuff and make new plans, or sometimes alter plans to make it better and have a better function. What will an genetic engineer do? An genetic engineer will directly manipulate the genes of an organism. So, the definition, the direct manipulation by humans of an organism's genetic material. This manipulation will mean work on the DNA, work on that tiny manipulation by humans of an organism's genetic material to change the genetic type of that organism. How? Insert the gene for insulin by a bacteria, and that bacteria will now produce the insulin. Alter the gene code that can increase the growth potential, let's say of maize, of cotton, and that will make a better food product. Or delete or inactivate a gene, like the ripening process of tomatoes. Buy a tomato, within a day or two, the tomato shrinked and is not juicy and does not taste like fresh. So, delete or inactivate that gene that cause the fast ripening, and you have a long life tomato. The definition again, grade 12. Genetic engineering is the direct manipulation by humans of an organism's genetic material to change the genotype. Genotype, those two little letters that tells us exactly what is the alleles, the two alternative forms of the same gene. Allele, two alternative forms of the same gene. How? By insert, by alter, or to delete or inactivate a gene. This brings us to a very interesting video that I would love you to watch, just to broaden your mind and understand where are we going to with this chapter.
Genetically modified organisms, or GMOs, have, as their name suggests, had their genetic character altered by humans. The notion of modification was developed by the founding father of modern genetics, the 19th century Austrian friar and scientist Gregor Mendel. Mendel studied plant variation, cultivated and tested some 29,000 pea plants, which led him to formulate the principles known as Mendel's laws of inheritance. In fact, humans have always crossbred both plants and animals to obtain hybrids with particular characteristics. In a broad sense, all domestic dogs descended from wolves are effectively GMOs. But in general, the term GMO applies specifically to organisms modified by modern biotechnology techniques. This usually involves implanting genes from one living organism into another, so animals or plants exhibit new properties, a process known as transgenesis. Researchers have, for example, taken a light-producing gene from a glowworm and implanted it into a cow embryo. While the future calf may not exactly glow in the dark, under the microscope its cells will emit a gentle glow under stress. Much used in research, genetically modified organisms have many practical applications, particularly in the field of agriculture and food production. Industrial companies have perfected varieties of plants like maize, soya and cotton, which are resistant to parasites, fungi and diseases, or to certain harmful herbicides used in farming. Other GMOs are designed to improve the nutritional value of crops or to have beneficial health implications. For example, American researchers are working on a genetically modified banana, which acts as a vaccine against gastroenteritis. But GMOs are also a cause for concern. While there's no concrete proof they're harmful, environmentalists say more research needs to be carried out into their possible health risks. There's also the worry genes in certain plants have a tendency to disperse and contaminate other varieties, thus upsetting the balance of ecosystems. Large multinational food processing companies have also rendered some organisms unable to reproduce, meaning farmers have to buy new seeds for every subsequent crop.
If you enjoy watching videos, that was a very good video to watch again, because it explains, give you the pros and cons. So watch it again if you like to look at videos and remember what you see on videos. Now this way of changing genes, altering genes and manipulating genes is not a new thing. Long ago, since the earlier days, humans manipulated genes. It was not like go into the DNA. No, but what we did is we breed specific plants together or specific animals. Those that come from a farm, you know that the farmer goes out and buy a specific bull because he wants to breed nice, meaty oxen. Now, and then you have another farmer that really wants to produce a high, good quality of milk. So he goes out and he is looking for a cow that really has the potential to give a lot of milk. And what about the bull? He's not giving milk. No, what will they do? They will look at his mother. Yes, and that was actually also the manipulation of the genetic material, although it was not direct. Remember genetic engineering? Direct manipulation by the human of an organism, genetic material. With selective breeding, we just had to take what we see in the phenotype, the physical observable features, and have the mother, have the father, and have a ratio or a probability of the offspring, and then pick the offspring after a few calves or a few plants grow. We can pick the one. But that was actually also manipulation of the phenotype, which in effectly is the manipulation of the genes, but not direct. The breeding of dogs. It all started out with a group of wolves that was manipulated, and that is something amazing if we look at all the different kinds of dogs. And then, of course, as I've said, specific type of cattle we know in Namibia, or to change the variance of colors and smells in flowers. that we can do. And look at this very interesting cauliflower. Now, colorful and manipulated. How will they do that? They do it with recombinant bacteria. That is one of the solutions. And this will be one where it's inserting a gene. In your textbook, you have a paragraph on that. It was a question in last year's exam. So please make sure that you know the steps. Where do we start? We start with the word plasmid. Do you remember that when we have the bacteria, very easy to draw. Now, special cell wall made out of peptidoglycogen, and inside there was only a string of DNA, and then there was a little circle that was called the plasmid. But that plasmid also contained genetic material. Inside this plasmid, the very clever scientists discover that to become antibiotic resistant in the bacteria, it was in that plasmid where that genes are being captured. And that put them on this pathway of using that plasmid. And what they need to do is they need to cut out a part of that plasmid, and they use E. coli. E. coli. It is Escherichia coli bacteria. Because they are a bacteria that is normally inside a human. When there are too many, you can have severe diarrhea, but they are actually inhabiting our inside. So it is not a foreign bacteria for the human environment, inside environment. So they cut out with endonuclease restriction enzyme. Watch that word. Quickly look at that word in your book. I want you to see it. I want you to see that paragraph. Good. They cut out a part. Then they go to the foreign DNA. The DNA that they actually want to insert into the bacteria. And then with ligase. I'm just writing it a bit bigger. With ligase, they connect that piece of gene into the plasmid. They put it back into the bacteria. And what do we know about bacteria? Every 20 minutes, they divide in two. Type of mitosis, exactly the same cells will be produced all the time. Mitosis. But we call it specifically binary fission in the bacteria when they mitotically divide. Now, so all the bacteria that comes after it has been implanted will be the same in a very short time. And now, whatever is on that inserted gene will be produced. That protein will be produced. And they can have a lot of that bacteria. Please familiar yourself with that paragraph on recombinant bacteria. So let's go through that again. Remove the bacterial DNA, which is in the plasmid. Cut the bacterial DNA with restriction endonuclease enzymes. Cut the DNA from the other organism with the restriction enzyme. Combine the cut pieces of DNA together with ligase. Ligase combined also the DNA string during the protein synthesis. Now, and DNA replication. Insert them back into the bacteria and reproduce the recombinant bacteria. The foreign genes will be expressed in the bacteria. What is the benefits? There are a lot, and you will see some more in the video. But bacteria can now start to make human insulin or the human growth hormone to help ill people, suffering people. Very interesting is that they also manipulated the bacteria to clean up oil spills, to help our ocean life to survive. And now we are going to look at the insulin. Insulin has a
condition known as hyperglycemia is fatal if untreated and leads to various symptoms such as excessive inflammation, fatigue, weight loss, and blurred vision. In order to control blood glucose levels, a hormone known as insulin is made by the pancreas to signal cells for the uptake of blood glucose. Type 1 diabetes is caused by insufficient blood insulin and is believed to be caused by a variety of factors, such as viral infection from the rotavirus and measles and genetics. In 1921, Dr. Frederick Banting researched a successful way to isolate insulin from pancreatic cells in dogs. They later began mass-producing insulin for diabetic patients from pigs, saving many. Porcine insulin and human insulin only differ by one amino acid residue at the C-terminus, in which the alanine in pig insulin is replaced with a threonine in human insulin. Because of this meant that pig insulin was compatible with human insulin receptors. While animal insulins are still sometimes used today, they must be very pure as there are impurities that have been shown to cause allergic reactions. In 1926, scientists first crystallized insulin, and then in 1955, a breakthrough by Frederick Sanger allowed scientists to sequence the DNA coding for insulin. The 3D structure was solved by Dorothy Hodgkin in 1969 through the use of X-ray crystallography. After Sanger's work, scientists could use this genetic code to synthesize their own insulin. Efficient mass production of insulin began in 1978 through recombinant bacteria, in which insulin genes were inserted for bacteria to transcribe and translate into insulin. Thus, the insulin that you use is mass-produced by lab bacteria.
To get the insulin from cattle and pigs, what a process did they go through? But what was interesting to me in the video, and that is why I used that specific one, is we know about the sequence of DNA and we know about that specific amino acid. And wasn't it interesting to see only one amino acid in this sequence that form the protein insulin differs from a pig and a human? But what is the problem? Using pigs, dogs, whatever. The first problem is, of course, the animal need to die to extract that insulin. That is the first problem. And the animal lovers do not like that. We all love animals. The second thing is the allergic reactions, which can cause a person to feel very ill or even to die. Could you remember what is an allergic reaction? An allergic reaction is where our body thinks there is a pathogen inside and it starts to react with fierce immune reactions and it sends out histamines. This histamines try to cut off that area to keep the allergen at one place. And then sometimes we have an itchy reaction to warn us something is wrong, or we cannot breathe because it is swollen, that area. That was an allergic reaction. A reaction to a normal substance that the immune system identifies as a pathogen. And the body tries to react to that. And therefore, because it's histamines that is secreted, we drink an antihistamine. Now that was a bit of revision. So the problem is, the animal need to die. Lots of animals need to be found to extract enough insulin for all the millions and millions of people that is suffering from diabetes. We also know that diabetes, especially type 2, which is a lifestyle disease, is increasing by the day. So more and more insulin is needed. And then the allergic reaction. So what is better? Make a synthetically insulin. The benefits is, it is human insulin. The benefit is, it can be produced in large quantities without any animal that needs to die. It's human, it's large, and it can be produced in a laboratory. So how will they do that? They take a cell in the human pancreas. Why the human pancreas? The pancreas receives the message that the insulin is low, the blood glucose levels are high blood glucose levels is received by the pancreas cell. Glucose levels, so it is the alpha cells in the pancreas, in the islets of Langerhans. There we got it. Nah. And this cells, alpha cells in the pancreas of Langerhans, receive the message, produce and secrete the hormone. So get that gene from a pancreas cell and cut it out with endonuclease restriction enzyme. Now, insert it into the plasmid. But first, get the plasmid, cut it with the endonuclease restriction enzyme, insert the correct genetic material, human insulin producing gene, and put it back into the bacteria. Recombinant DNA, important word. It means it is recombined. To combine, recombined. Now, it was combined, changed, and combined again. Put it in a fermentation tank where it is the perfect conditions for the bacteria, food, warmth, correct temperature. Now, and now it starts to produce the insulin, multiplying. Of course, which type of cell division? Mitosis. What do we name the way bacteria reproduce? Binary fission. And eventually they can extract the pure insulin. Amazing human insulin. Can you see, grade 12s, how many of our previous chapters we can now put into this new chapter because we know so much. Please complete your worksheet attached and then you will be good for our exciting next session.
you're the song of my heart. Mama, need I tell you that I love you? Mama, need I tell you that I miss you? You're my wings in my eagle. You're my shadow in my sunlight. Mama, need I tell you that I love you? Mama, need I tell you that I miss you? You're the queen of my world, you're the sound in my heart. Mama, need I tell you that I love you? Mama, need I tell you that I miss you? You're my wings in my eagle. You're my shadow in my sun. Every day's bright memories. Mama never ever let me down. Legacy is what you left behind. Biba, 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 and your love will never fade. Life never gonna be the same. Everything happens for a reason. Jaja man bless every woman. Jaja man bless every mother. Father God, thank you for your love. Biba, 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 hey. Mama, need I tell you that I love you? Mama, need I tell you that I miss you? You're my wings in my eagle. You're my shadow in my sunlight. Mama, need I tell you that I love you? Mama, need I tell you that I miss you? You're the queen of my world, you're the sun in my heart. Mama, need I tell you that I love you? Mama, need I tell you that I miss you? You're my wings in my eagle.
Now this is what's the year what the line get. You make sure I catch your bar on the end day. And Jila down go dapa to Luca. 80 or dapa to Luca. Mambo kasi where moyo. Me you the queen of my heart. Since from the start, yes you must stop. But you're in our part. I'm the president of the cool hole. I'm born to she go to I went through a lot of bad this ways, but you made sure you were there for me. Yeah, I went through a lot of darkest ways, but you made sure you were there light for me. Me manda lukale na me feel figu hadi kienda kengolo. Yeah, manda lukale na me feel figu hadi kala kandi wende.